A very big hello to all my wonderful PU2 champions. I hope all my dear champions are doing absolutely good. And today I have picked up a new chapter, that is molecular basis of inheritance, which we have not touched this year. So this is the first time, and my dear children, the reason why I am picking the high weightage chapter first is that by the time of examination you would have enough time to revise these chapters that we are studying now. So mostly children, what they do is that they pick up some easy chapters so that it builds up their confidence, and then they move on to high chapters. Sometimes they do not get time to complete the high chapters. They think it's difficult and they then just leave it. I do not want you to be one among them, because this way is partially correct and partially incorrect. Why? I'll tell you. Because my dear children, sometimes you spend too much time on the easy chapters. Now if at all you're picking up a high weightage chapter at this particular point of time and you still have some time for examination, the good part of the story is that yes, I am finishing it and then I have time to revise it while I study the new chapters as well. In that way you are most safe, my dear children. And the problem with picking up a high chapter usually is that you feel it's tough, so you give 4 days time or 5 days time to complete the whole chapter. Don't do like that. If you give yourself a limited time, you will try to finish it as soon as possible in a better way as well. And the best part of the story is that I am here with you and I am telling you we can complete molecular basis of inheritance, which is the second high weightage chapter that we are having, within a single day. How? By watching the one shot video where you understand the concepts, then solving the 1 marker questions, and then solving the 2 and 5 marker questions that have come all these years, my dear children. So don't you think your chapter is done? Now the next day you can include one more day for your preparation where you do your study about this particular chapter, and then it's done. Within 2 days this chapter is done. You get it? And you have to use this strategy. It is going to definitely help you out. If at all you think whatever I spoke makes sense to you, what you have to do, click on the like button right now, and you can also let me know in the comment section. And my dear children, if you love or like the way I teach, you have to let me know. If you do not like the way I teach, you have to let me know. If you have doubts, you have to let me know in the comment section. I am here for all of this. Okay? So starting with the weightage of the MCQs. So every single blueprint that you see, my dear children, you see one MCQ has come in each blueprint out there. So definitely one marker question is gonna come, which is why we will have a quick practice of few questions, at least 20 of them. I'm not taking it to 30 so that the video is not too long for you to study. We'll restrict it to 20 and I'll explain you the concepts behind all the questions. Agreed? Done? Deal? If you are in, let me know that. Now, the number of nucleotide pairs present in the DNA of the primary oocyte of a newborn in human is. You are asked about the number of nucleotide pairs, as in you're talking about the length of the DNA and everything, okay? Or the number of nucleotides directly. In what? In the primary oocyte of a newborn. In a newborn, the primary oocyte of a newborn, you're asking what's the number of nucleotide pairs. So generally what we think is that primary oocyte is a stage in oogenesis. So probably the cells might be undergoing meiosis, and whenever we say meiosis is happening, my dear children, what happens is that the 2N, the diploid nature which is there, the cells which are in diploid, they becomes haploid, isn't it? They become what? Haploid. So we expect this to happen. Okay, so we expect this to happen. So if 2n number of nucleotides means, if at all it is 6.6 into 10 to the power 9, then we expect haploid things to be 3 to the power, 3.3 into the power 10 to the power 9, that is half of it. But the primary oocyte, has it completely undergone meiosis to be haploid, or is it actually diploid in nature? It is diploid. And how do you know it? If you study the human reproduction chapter properly, you will definitely know it. So over here, my dear children, the primary oocyte has just started off the meiotic division, has not completed it. So it is in diploid condition, and this is the number of nucleotide pairs over here. So 6.6 into 10 to the power 9 would be the answer. If at all you are asked what is the number of nucleotide pairs, what is the number of nucleotide pairs in a secondary oocyte, then it would have been, what my dear children, it would have been half. Why? Because the primary oocyte undergoes meiotic division, becomes half in number, the number of chromosomes becomes half, or the nucleotide content becomes half, and that happens in the secondary oocyte. So the question is asked like this, you have to pay attention to the details out there. So I hope this is paka clear and you will be able to answer this question if at all you see it coming like this anytime soon. Now the second question, a DNA segment has a total of 1000 nucleotides. 1000 nucleotides. out of which 240 of them are adenine containing nucleotides. How many pyrimidine bases in this DNA segment does this DNA segment possess, my dear children? So you are given such information, that is 1000 nucleotides are there in total. Okay, this is the total number of nucleotides, out of which you have said that 240 of them, just a second, whatever I am thinking, I am not able to write that. Why? 240 of them are what? Adenine. Now adenine is what, my dear children? Adenine comes under purines. So in nucleotide bases you have two types. One is purines and the other one is pyrimidines. A and G are purines. A, G, O, G we say no, like A and G. A base pairs with the pyrimidine, that is thymine, and G base pairs with what? Cytosine. So if at all it is said that 240 adenine is there, that means how many pyrimidine will be, how many thymine will be there? A is the one that base pairs with thymine. So whatever number of adenine is there, that many number of thymine will also be there. 240. Agreed? Done. Now in total, how many nucleotides are there? 1000. So 240 plus 240 is equals to what? 240 plus 240 is what? 480, my dear children. Now in total, how many do we have? We have 1000. In total, we have 1000. Now if you do the calculation, what do you get? So over here, you have what? Now I do a very what do you call a different type of calculation. So please, you do not be scared about any of it. So you get 520. Okay, you get what? 520. Now this 520 which is there, you have to divide between both of them. Agreed? You would have to divide between both of them, because guanine plus cytosine should be 520. So 520 divided by 2 of them would be giving you what? 260. So I'm assuming 260 guanine is there. If guanine is 260, then obviously cytosine also will be what? 260 in number. Now what are you asked, my dear children? You are asked, how many pyrimidine bases does this DNA segment possess? So pyrimidines are thymine and cytosine. So what do you have to do? You have to do this plus this. 260 plus 240 is equals to what, my dear children? It would be 500. And your answer is B. Do not go by this calculation, okay? Okay, so anyways, it is 520, and 520 divided by both of them, that is 2, is equals to what? 260. And then if you calculate the number of pyrimidines, you get it to be what? 500. I hope whenever these type of questions come, you do the calculation like this. Usually what do we do is that, okay, we got 260. Now 260 plus 260 is what you do. No, they are asking specifically about pyrimidines. So you should know it is this. Cool? Works? Are you liking these type of questions? It's fun, no? Doing these type of questions? Now, going ahead, given below is a portion of DNA strand giving the base sequence on the opposite strand. What is so special shown in over here? So 5-GAA TTC 3-. And then over here, ulta is there, 5-GAA TTC 3 is there. What is so special? Does it show replication completed? Does it show deletion mutation? Does it show that there is a start codon at 5-? Start codon is AUG. You do not see AUG anywhere. Palindromic sequence, what do you see? What is so special? So it is palindromic sequence. What do you mean by that? So palindromic sequences are the one that you can read from here to here in the same way you would be able to read from the opposite direction. So 5 to 3 you read it as GAA TTC, and over here also from 5 to 3 you read it as GAA TTC. Okay, so they are what? Palindromic sequence. This would be your answer. D. Perfect. Okay, you cannot say replication is completed or not. You do not give, they have not given the whole thing and they have not mentioned anything like that. How can you say the replication is completed or not? This is just a segment, right? Okay, cool. Now going ahead. The 2 nucleotides of DNA are linked to 3 and 5 carbon of pentose sugar by. So my dear children, the pentose sugar which is there. 1, 2, 3, 4 and 5th carbon is over here. Similarly, you will have another pentose sugar. 1, 2, 3, 4 and the 5th carbon is there. So now over here it is said that 2 nucleotides. So this when it attaches to a nucleotide base over here, similarly over here also nucleotide base over here, and over here there is phosphate group attached on the 5th carbon. Okay, so this is one nucleotide and this would be another nucleotide which has one pentose sugar, one nucleotide base and also one phosphate group. So it is said that when 2 nucleotides of the DNA are attached through the 3rd carbon and the 5th carbon, my dear children, what do you call this bond? So when one carbon attaches to the phosphate group, this bond which is there, you call this as phosphoester bond. Right? We studied that phosphoester bond. Similarly, here also carbon is attaching to the phosphate group, leaving some groups out there. Okay, so this is also phosphoester bond. 2 phosphoester is there. So what do we call it? Phosphodiester. 2 is there, 2 bonds are there, so phosphodiester, my dear children. This is phosphoester, this is also phosphoester, so phosphodiester would be the correct answer for this question. Mostly children go and attempt phosphoester bond, wrong. They are asking about 2 nucleotides, what is there between here and here. So both of them you have to say. If they asked you what happens if the phosphate group attaches on the 5th carbon of the pentose sugar, what do you call this bond? That is phosphoester bond. If you are asking about both of them, it is phosphodiester. Cool? Next question. In biochemical characterization of transforming principle, which of the following enzymes affect transformation? So when we say transformation, my dear children, you might have seen the experiment which was done by Avery, MacLeod, McCarthy. Okay. Over there, you saw that whenever certain enzymes were being put down, they were treated with certain enzymes, the transformation of what do you call the smooth one, sorry, the rough one to smooth one was not taking place. Okay, so what was that enzyme in which this transformation was not taking place? When they added protease, does the transformation take place? Yes, because proteins were getting damaged and they didn't have much to do with the transformation. When they added amylase, was transformation taking place? Yes. The carbohydrates were getting damaged, but that also didn't have much things to do with the transformation. What about lipase? Lipase is for lipids, that is fats. So the fats were getting damaged. Still, they didn't have much to do with the transformation. But when they treated with DNAs, the DNA was getting damaged, my dear children. And when DNA is getting damaged, do you think this transformation can happen? No. Okay, this transformation was not happening. From from rough to smooth transformation, it was not happening when it was treated with DNAs. And this was the experiment which was done by Avery, MacLeod and McCarthy, and they said that this is probably the thing which is acting like a transformant, or this is the one that causes the transformation. DNA is the one that causes transformation. But no one believed them, which is why some other people had to come and do the experiment for that. And tell me in the comment section, who are the other people who said that no, whatever they said was actually correct. So you have to tell me, and that was the last experiment to prove that DNA is the genetic material. Now the 6th question, the experiment that proves the semi-conservative principle of DNA replication. What do you mean by semi-conservative method of DNA replication? Such that, my dear children, suppose this is the DNA of the parent. Now over here, this strand separates out, this strand separates out, and you get a new strand over here complementary to this, again a new strand over here complementary to this. But if you see, in the new DNA which has been formed, one is the parental one and the other one is the new one. So don't you think there is some conservation going on over here? The DNA is still being conserved over here. So but it is not fully. Both of them are not parental. One is new, one is parental. So this is semi-conservative mode of replication. What do you see? Generally see. Now who proved that this was happening? It was what? Is it Griffith's experiment? No. It is Hershey and Chase's experiment? No. For those of you who answered the previous question, you are absolutely correct. Hershey and Chase were the one who gave the final testament that yes, DNA is the genetic material with their experiment on bacteriophage and E. coli. Okay, anyways. Avery, MacLeod and McCarthy experiment? No, I told you they were trying to prove that DNA is the genetic material. Again, Michelson and Stahl experiment. Yes, they were the one who said that DNA is replicating semi-conservatively. So over here the strand was there, okay, which was made in a medium which had N15 isotype, okay, of nitrogen, my dear children. And then later on they changed the medium to N14, and when they changed the medium to N14, my dear children, the new strand which was being formed over here had N14, whereas the older one which you see in blue, it was made up of N15 itself. So with this they got to understand that yeah, something is different. So when they centrifuged over here, they were able to find that all of them were settling at a safe place, and it was heavy. In centrifugation they were settling all down. But when this replication happened, and after that the centrifuge, they found that okay, all of them were settling somewhere in the middle, okay, in the hybrid region, my dear children. So they thought that there was some changes out there. Again they tried to replicate, and then they were able to find out, my dear children, that you get a new set of brand also. The ones that are purely made up of N14 only, and the ones that would be hybrid. So there is semi-conservation happening, which is why you have this strand going on and again and again and again and again. You get it? So this was measles in a stall experiment. I have explained this to you in the one-shot video properly. Please watch the one-shot video if at all you are not able to understand. Okay, fine. Chalo. Next. Average rate of polymerization during DNA replication is what? The average rate of polymerization during DNA replication is what? So for eukaryotes, it's very less, but for prokaryotes, it's very high. Rate of polymerization means, as in, for example, the nucleotides to come together to form a polymer of nucleotides to form the DNA strand. The average rate of polymerization during the DNA replication process is what? Please remember, it's given in the NCERT. Some people do not tend to focus on this part, but the answer is 2000 base pairs per second. Okay, per second 2000 base pairs are being added up. If you have seen DNA replication in E. coli, they completed within 18 minutes. And that is so strange, right? Within 18 minutes, they are completing the whole replication. Why? Because their average rate of polymerization is what? 2000 base pairs per second. Okay, moving ahead to the next question. Question number 8. GGU, GGC, GGA and GGG all code for glycine. Which salient feature of the genetic code does it prove? What do you think? All of these codons, my dear children, if you see, all of them are unique. Over here it's U, over here it's C, over here it's A, over here it's G. All of them are different, but yet they are coding for the same amino acid that is known as glycine. So does it show unambiguous nature of the genetic code? What does unambiguous mean? Unambiguous mean one codon is gonna code for one amino acid. So if at all the question was GGU is coding for glycine and no other amino acid, which salient feature does it show? So then your answer would have been what? One codon, one amino acid. That means it is unambiguous nature that is being shown over here. But over here 4 codons are given, and yes, it is said that they are coding for glycine. That means if 4 codons are coding for the same amino acid, or instead of 4 codons, if I write multiple codons are coding for the same amino acid, what feature does it show? It shows something that is known as degeneracy, or they are known as degenerate codons. So all the codons think that, okay, GGU thinks, okay, I am glycine's best friend, no one else. But then later it comes to know that no, GGC is also its best friend. All of them are coding for what? Glycine. So this is degeneracy, and this feature is known as, this is what you call as degenerate codons, my dear children. It's not universal codon. If at all it was said that GGG is coding for glycine in human beings and GGG is coding for glycine in E. coli also, then in that case it would be universal nature that is being shown. That means one codon is universally coding for one particular amino acid, that is universal codon. Initiator codon has nothing to do with this. Initiator codon is the one which helps in initiating the process of translation, my dear children, and it has nothing to do over here. So AUG is also an example of initiator codon, which is nowhere mentioned up over here. Okay, so your answer for this question would be what? Degenerate codons. Now let's move on to the next question. Amino acylation of tRNA. What do you mean by amino acylation of tRNA? On the tRNA, when an amino acid comes and attaches, my dear children, this process is known as amino acylation, or you also call this as charging of tRNA. Now, when do you think this occurs? When do you think this is needed? tRNA, amino acid is needed when the process of translation is going on. When whatever is there in the mRNA, this information, this codons, okay, this is the codon. On the codon, one anticodon comes and attaches. Okay, and the tRNA which has this anticodon, that it also has the amino acid. So this codon is coding for this amino acid, and this process of translating the information of mRNA to form protein is known as translation. So your answer for this question would be what? Translation. And this is the first step of the whole process of translation. Always remember, amino acylation of tRNA. Okay, it happens in the first phase. Yes, the next question. The coding sequences present in the processed RNA is, what do you mean by processed RNA? So RNA, where, my dear children, in RNA there are some regions that codes for proteins, okay, and there are some regions that do not code for proteins. Some region that codes for protein, some region that do not code for protein. What do you call these regions as? You call them as introns. Okay, the one that intervenes, the one that does not have much work to do out there. And what do you call them which codes for proteins? You call them as exons. Okay, now after the processing of RNA, these introns are removed, my dear, in the way that you get only exons. You get only exons at the end, so that you just have things that codes for protein, nothing else. So the coding sequence present in the processed RNA is what? Is it exons, introns, cistron, genes? It would be what? Exons. Gene is a segment which contains all these things. Cistron is again a segment coding something that would be coding for a protein. So it's all talking about segment. But over here you are specifically saying that this is the region that codes for a protein. Okay, or the coding sequences out there. So your answer would be B. Now, if a processed mRNA of a particular cistron containing 636, listen, cistron, okay? If a processed mRNA of a particular cistron containing 636 base pair that codes for a polypeptide chain, what are the number of amino acids that it codes for? If you are given that 636, okay, if it looks like something else, I'll just remove it. Okay. You will count as 6 only. So 636 base pairs are there. You have to find out how many amino acids are going to be made from this thing. Always have this understanding. When you're talking about an mRNA, the first codon will be start codon. And that actually codes for what? And that actually codes for amino acid, that is methionine. That's totally fine. It will be coding. But the last codon which is there, 1, 2, 3 base pairs which would be there, they are stop codon. They constitute the stop codon. They does not code for anything. So if we say 636, you know 3, it comes in a group of 3. You know it comes in a group of 3, right? All the codons comes in a group of 3. So if we calculate, it would be what? 2, 1, 2, right? It would be 2, 1, 2 amino acid. But the last one is not actually coding for anything. So we can do -1. So this won't be counted as a amino acid since it is not coding for anything. So if I do -1, what will be my answer? 211. So your answer would be 211. Okay, got it? So 636, you're asked how many amino acid will it form. So my dear children, they have not mentioned the stop codon is there, but you know that in the processed mRNA you have start codon, you have stop codon, you have everything like that, and they do not code for anything. So the last 3, they will be coding for 1 amino acid only. So at the end of the whole process, you just minus 1 amino acid, and that's what you get. That's when you get your answer, that is 211. I hope it's crystal clear. Perfect? Yes? Okay. Now the next question. In pea plants, each character is governed by a gene which is monocistron, polycistron, operon, intron. What do you say? In pea plant, pea is basically a plant, it is a eukaryote, right? Eukaryotes are the one that has a definite nucleus, double membranous cell organelles, all of it, right? In the case of eukaryotes, my dear children, we have monocistronic genes. That means over here there will be a gene that would be coding for one particular protein. When you talk about prokaryotes, they have polycistronic genes. That means if you talk about this segment, this will code for protein 1, this will code for protein 2, this can code for protein 3. So you get multiple proteins, okay? You get multiple results out of this particular gene segment. But that's not the case of eukaryotes. They just have one cistron that codes for what? One protein. Okay, they just have one region that codes for one protein, single cistron. Okay, over here they have multiple cistrons, they code for multiple proteins out there. So in pea plants, which is if at all you do not know that pea plant is basically a eukaryote, you will not be able to answer it. If you don't get that catch of the story, you will not be able to answer it. But since you know now, you will be able to answer. So your answer is monocistron. Okay, cool. Next. Which of these are correct? UTR, untranslated regions, are located on DNA. Okay, UTR is present both at 5' before the start codon and at 3' after the stop codon. UTR provides the stability to mRNA. UTR is required for efficient translation. What is UTR? UTR is untranslated region, my dear children. Translation happens in what? Translation happens in the mRNA. So this UTR is not found in the DNA, instead it is found in what? The RNA, my dear children, always remember. UTR is found in the RNA. It is present before the start codon and after the stop codon. Ma'am, what's the use? You know, in our body there are so many enzymes that are there to cut certain things out there. Exonucleases you call them, okay? They cut from the ends. So you want to protect the mRNA that is going to code for the protein from all these things. That is why you have UTR. It provides stability. It is also required for efficient translation. So UTR and mRNA, which are their best friends, you have to think like that. Okay, so UTR, untranslated regions, are located on the DNA is a wrong statement. This statement itself is wrong. You are asked about the correct one. UTR is present both at 5' end before the start codon and also at 3' end after the stop codon. Correct, ma'am. UTR provides stability to the mRNA. Correct, ma'am. UTR is required for efficient translation. Correct, ma'am. So 2, 3 and 4 are correct. So your answer would be D. Got it? Okay. Now, if the template of DNA strand is 3 ATGCA8TCT 5', its complementary DNA strand would be what? Tell me, what will be the complementary DNA strand of it? So over here it starts from 3' and ends in 5'. So the complementary DNA strands will end with 5' and end with 3'. Start with 5' and end with 3'. Over here ATGC is given. So opposite to it, what do we have? A base pairs with T base pairs with A, G base pairs with C base pairs with G. If you see this, let's compare. If at all we are not getting the answer with doing this much only, we can do more. Okay. So 3' wrong. 3' wrong, because the complementary start will be having 5'. So 5' TACG, okay, this is correct. 5' ATGC, it's wrong. So with this statement only we were able to find it out. But my dear children, please be cautious. Whenever needed, you have to fully decode it. Okay, so if at all TACG is correct over here, but the rest of the things are wrong, then what will you do? So please make sure you're going through it properly, the options properly. Okay, and if at all you're asked about RNA, they do not have what? Thymine, they have uracil. Always remember that as well. So your answer is C over here. Now going ahead with the next thing. Match the following: Alec Jeffrey, F. Sanger, Jacob and Monod and Griffith. I always tell whenever you're solving match the following question, go through these things first. First side once and then the second side. You will get to understand a lot of things. You will be able to spot the easier ones. So Lac-Operon, transforming principle, annotated DNA sequencing, DNA fingerprinting. With my knowledge, I'll tell you something. Griffith, we all know he was the one who did the transforming principle experiment, right? With the mouse and the pneumonia and things like that, right? Pneumonia, Streptococcus pneumoniae. So over here, D goes with 2. D goes with 2. What all options do I have? I just have one option where D goes with 2, and with that I can tell the whole answer. So Jacob and Monod also, we know that Lac-Operon is made by them only. Jacob and Monod. So C goes with 1. Now F. Sanger and Alec Jeffrey's list, most of the children are getting confused in that. So Alec Jeffrey goes with DNA fingerprinting, and F. Sanger, my dear children, he goes with annotated DNA sequencing. Okay, always remember. So here F. Sanger, S is there, here also in DNA sequencing S is there. You can remember like that if at all you want. And Alec Jeffrey, and over here you have DNA fingerprinting. Okay, cool. If A and B are the DNA of two mothers, A and B are the DNA of two mothers, and C is the DNA of the child, on observation of banding pattern of the DNA fingerprinting, it can be concluded that what? Can be concluded what? So this is A and B, and this is the child's one. So if at all you see, C and B are almost matching in all senses, my dear children. So probably what, my dear children, according to DNA fingerprinting, B might be the mother of C. So C is the child of A. C is the child of A? No. C is the child of B? Yes. It's the same thing. Mother, B is the mother of C, or C is the child of B, the same thing. So this is your answer. C is not the child of either A and B, wrong, because you have the same type of pattern over here. And relationship unclear, C shares bonds with both. No. Your answer is B, because here you can clearly see all the strands are basically matching. Okay. There would be obviously certain changes from mother to child, my dear children. It's not like always it will be there, but most of it will be definitely matching in the DNA fingerprinting. Okay. Now, okay. During transcription, the site of DNA molecule at which DNA polymerase binds is called. Tell me. During transcription, what would be your answer for this question? during transcription the site of DNA molecule at which, just a second, you have to form RNA, right? So RNA polymerase, not DNA polymerase, okay? During transcription from DNA you make the RNA, right? It's mRNA. So RNA polymerase binds is called, what do you call that site? Promoter, regulator, enhancer, inducer. Your answer is promoter region. It will be binding onto the promoter region, okay? Always remember it. Cool? Easy question, no? Yes. Now next, in a DNA strand, if there are 100 nucleotides, again 30 are adenine. Now this is the other way of asking the same question that we solved before, okay? And the number of thymine, guanine, cytosine respectively are. So this time you are not just asked about pyrimidine, you are asked about one purine also. So you have to talk about all of them. So if A is 30, T is also 30, agreed? Because A base pairs with T. Now if both of them are 30, 30, my dear children, so total is what? 60. Now out of 100, 60 are gone in them only. So your answer is 40. Now 40 is there, you have to divide between 2, that is guanine will be 20 and cytosine will be what? 20. Agree? So now you tell me. Thymine first. So thymine is what? 30. 30. So these two options are there, this and this. Let's leave these two options. Guanine 20 and cytosine also 20. So your answer would be this, the first one. Cool? Okay, now let's move on to the next question. In Hershey and Chase experiment, which radioactive isotope was used to label the DNA? So over here, the term DNA is being mentioned. You want to radio label the DNA. So first of all, you have to think of what is DNA made up of. It is made up of nucleotides. What is nucleotide made up of? A pentose sugar, a nitrogenous base and a phosphate group, right? So if at all we want to radio label the DNA, we are going to radio label something of this particular part, either the phosphate group or the pentose sugar which is made up of carbon and hydrogen, or probably the nitrogenous bases which has so many things around it. Okay, the nitrogen is the amino compound and all of this thing, right? You have to target any of these things. Now in these options, C and H is given, so ma'am, carbon and hydrogen is given, but in Hershey and Chase experiment, these guys were not being used. Because these are common to what? Proteins, as well as in protein also you have carbon and hydrogen, as well as in the case of DNA also you have carbon and hydrogen. So I cannot use it. I have to be very specific out there. So out of them, phosphate and sulfur, which one is the unique one that is found in the DNA? It is the phosphate. So to radiolabel DNA, which isotope will I use? I will use phosphate 32. If I wanted to radiolabel the proteins, I would use sulfur. For proteins, I would be using sulfur, and that was actually being done by Hershey and Chase in their experiment as well. Okay, do not forget it. Always remember S35. So 5 looks like S, right? So do not forget the isotope's name. Now going ahead. Human insulin is produced in E. coli cells by rDNA technology. This proves that genetic code is what? What does it prove? So human insulin, which is actually our insulin, humans insulin, the same thing is being produced by E. coli because we are doing some rDNA technology, recombinant DNA technology out there. We are changing some stuff up there. Still it is being able to produce it. What does this show? That the genetic code which is there, the codons which are there, my dear children, what does it show? They show that it is universal. It's the same up there. Whatever this particular codon is coding for this amino acid, probably the same codon will be producing the same type of amino acid in other organisms as well, which is why we say the genetic codes are basically what? Universal. There are certain exceptions to that, but mostly they are universal. So your answer would be A. Okay. Now I know there might be so many concerns still lingering, where you are not doing that great. Do not worry, I will be covering all those concepts in the 2 and 5 marker section. So be there. Okay, the video will be soon published just after this video ends, and take out some of your time and revise whatever you have studied until now. It is going to help in better retention. And if at all you think that this video had helped you in some ways, what do you have to do? Click on the subscribe button right now, click on the like button, and also let us know with the help of the comment section below. Okay, wishing you all the best. Take care of yourself. Lots and lots of love from Jasmine. Bye bye.